Cloning is the process of creating genetically identical copies of an organism, and it can be done in both plants and animals through various techniques. In plants, cloning can be achieved by two primary methods: tissue culture and cuttings. Tissue culture involves taking a small group of cells from a part of a plant, such as a leaf or stem, and growing them in a nutrient-rich medium under sterile conditions. These cells can develop into new plants that are genetically identical to the original plant. This method is particularly useful for producing large numbers of plants with desirable traits, such as disease resistance or enhanced growth, in a relatively short amount of time. Cloning plants from cuttings is a simpler technique where a section of a plant, usually a stem or leaf, is cut and planted in soil or water. This cutting will develop roots and grow into a new plant that is genetically identical to the parent. Cuttings are commonly used by gardeners and farmers to propagate plants that have desirable characteristics, ensuring that the new plants will be true to the original. Animal cloning can be more complex, with one common method being embryo transplants. This process involves taking an embryo at an early stage of development and splitting it into several individual cells, each of which has the potential to develop into a separate organism. These cells are then implanted into the wombs of surrogate mothers, where they grow into clones of the original embryo. This technique is often used in livestock breeding to produce multiple offspring from a single, genetically superior animal, allowing farmers to rapidly increase the number of animals with desirable traits such as high milk production or lean meat. Another more advanced technique for creating animal clones is adult cell cloning, also known as somatic cell nuclear transfer, SCNT. In this method, a nucleus is taken from an adult cell of the organism to be cloned, which contains all of its genetic information. This nucleus is then inserted into an egg cell that has had its own nucleus removed. The egg, now carrying the genetic material of the donor organism, is stimulated to begin dividing and developing into an embryo. This embryo is then implanted into a surrogate mother, where it develops into a clone of the original adult organism. The most famous example of adult cell cloning is Dolly the sheep, who was the first mammal to be cloned from an adult cell in 1996. This breakthrough demonstrated that it was possible to create a genetic copy of an organism from a single adult cell, raising the possibility of cloning animals with specific genetic traits. Cloning, however, raises numerous ethical, environmental, and practical concerns. One of the main issues is the potential for reduced genetic diversity. Cloning produces genetically identical organisms, which can lead to a lack of variation within a population. This makes the population more vulnerable to diseases or changes in the environment, as there is less chance that any individuals will possess traits that could help them survive. In agriculture, for instance, a disease that affects one cloned plant could easily wipe out an entire crop if all the plants are genetically identical. There are also ethical concerns, particularly regarding the cloning of animals and the potential for human cloning. The idea of cloning raises questions about the moral status of clones and whether it is right to create living beings for specific purposes, such as organ harvesting or experimentation. Additionally, the cloning process itself can lead to high rates of failure, with many embryos not developing properly or resulting in clones with health problems. This raises concerns about the welfare of cloned animals, as the techniques used can result in suffering and early death. In summary, cloning is a powerful technique that allows for the production of genetically identical organisms, with applications in both plants and animals. Plants can be cloned through tissue culture and cuttings, while animals can be cloned using embryo transplants and adult cell cloning. However, cloning raises significant ethical, environmental, and practical concerns that need to be carefully considered. These issues include the potential for reduced genetic diversity, the welfare of cloned animals, and the broader moral implications of creating cloned life forms.